Christine, welcome. Okay, I'd like to introduce Professor Christine Beveridge, the Director of the Australian Research Council Centre of Excellence for Plant Success in Nature and Agriculture, and she is the chair of this session, Fred.
Thank you. I did misread the message. I was thinking it was the next session, physiology, because I'm a physiologist, but I also do a lot of phenotyping, so I'm also happy to chair this session. And I'm really proud of this meeting, and Mark and Fred and Charlie and others have been running over the last few years, and I really value the fact that we're integrating across all different areas of prediction in the G/E/M space. And I wanted to reiterate points that people have said before about breaking down the silos. So with no further ado then, I'll introduce the first speaker. I'm very proud to introduce this speaker because it's Danielle. So where's Danielle? There we go. Thanks. Come up and let's get started in predicting G/E from trait dynamics. Danielle.
I think my where is the pointer? Okay. Yeah. Thanks for the introduction, Christine. It's a pleasure to be here after almost, well, I moved to Chile, back to Chile last year. So now I want to show a little bit some of the work that we have been doing there with a team of researchers from different universities and from INEA, that is a government-funded institute. So I am sitting in the south of Chile right now, in Valdivia, so it's a very green city. People think that Chile is only desert. No, we are green in the south. It rains about 3 times more than in the Netherlands, so but more focused on during the winter. So we have sunny and more dry summers. And we have good beer if you want to visit. So we have been talking a lot about the target population of environments. This is a sort of recap of what we are trying to do. So we have this set of environments that are interested for breeders, interesting for farmers, and we want to represent them by a number of locations, years, and managed environments like Professor Pifo was illustrating this morning. We try to select those years and locations as good as possible so that they can inform what is happening at the farmers' fields. So here, again, what we are trying to do, we use this sample of the target population of environments that is represented by these multi-environment trials, and we also have a sample of our germplasm, and we test them, and we hoped that this set of multi-environment trials can help us predicting what will happen in new environments or new genotypes, or the most challenging ones, new genotypes and new environments. And there, well, I will not go into details again because this was discussed this morning, but basically what to try to do is to find out how we can increase our genetic gain, introducing predictions and also, like I will try to do in this talk, introducing new phenotypic methods. So I think, well, this paper came out a few years ago, some discussion. I think there was a phenotyping symposium in Mexico, and then you see all sorts of names here, and we have a challenging and very exciting afternoon trying to discuss what are we going to do with all this phenotyping that is coming up. Because nowadays, well, you have MPAC, the Australians have many drones, and Zurich also, you have many nice phenotypic information. But how we use that for prediction. So at the beginning, we start with something, many pictures, many pixels, but each of those pixels is not very predictive. So we need to do something to correct for what is, on one hand, how we can go from this picture to a number. That is informing best what is happening each experimental unit per time point, but then we need to correct for spatial effects, trends in the fields, and later, over time. And the question is how we can incorporate this time dimension in those prediction models. We usually work, or the most common model is like GBLAP, for example, that people have as a reference, so how we could expand that model so that we can incorporate all those dynamics. So here is a cartoon way of looking at it. So we start from raw phenotypic data. So each of these slices represent one moment in which our trial was measured. Then we try to separate. So each little dot here represents one plot, one experimental unit. And we need to separate what is happening from this spatial trend and what is happening with the genotypes. That at the end is what we want to do when we do breeding. So after we have separated this, we extract those blues and we try to represent what is happening over time. So this is just a cartoon example in which we have two genotypes that are very contrasting for their response to drought and heat. For example, here we have genotype 1 that when we change water availability, the dynamics don't change so much. So if we would extract the parameters that are representing or describing those curves, well, those parameters would be the same, irrespective of the water level, or almost the same. The opposite is true for genotype 1 and different types of temperature conditions, because when we change temperature, the parameters change a lot. So in that sense, we go from something that is time-dependent to something that something is the parameters that describe this curve that is summarizing what is happening over time, and that is describing G/E, because we have another genotype that has the opposite reaction. So when we change water, parameters change. When we change temperature, parameters don't change. So that's representing and already summarizing G/E over time. And later, when we extract those parameters, we can model the change in those parameters over time in response to water, temperature, or whatever stress is interesting for our crop. And if we know that, we can, in principle, predict the whole, what we call the adaptation landscape. So to describe for any type of combination of weather or environmental driver, how the genotypes would react. And, well, a little bit like Fred presented this morning with these surfaces. So if we can know that genotype 1, okay, increases the yield when we change temperature or water or whatever, we can know for each combination which genotype would do best. That's in theory. And that was before I went to Chile, so I was sitting nicely in my computer and getting nice data. In Spanish, we say otra cosa es con guitarra. Another thing is when you have to play the music. So because now I started to understand why they were looking at me a bit funny when I say, you need one extra replicate, you need one extra year, when you need one extra location. I moved to Chile with this nice picture in mind, and I said, well, let's do some trials. And I was very lucky because I was interacting with researchers that already had experience doing trial. But nevertheless, when you need to go with a drone and train your team and to do the things, I think it's a lot more difficult to have these very beautiful curves. So let's see how it looks like for a case study that I applied in Chile together with the Universidad de Talca, Universidad de la Frontera, and INEA. So you see, there is a very large set of devices now you can measure. There are very nice sensors you can use for phenotyping at different scales. Well, what we did, well, actually, we started from some previous research that was done by the group of phenomics led by Alejandro del Pozo from Universidad de Talca, and they had studied this stay green trait. So it's basically trying to describe. So here we have flowering somewhere, and then after flowering, well, the genotype starts to senesce, so they start to become more yellow. So the idea is that those that are able to stay green, that senesce a bit slower, they can keep up with a nicer, with more photoassimilates for grain filling, or therefore they should be able to sustain their yield level when they're exposed to droughts. That's a bit the philosophy. So they had studied, in this case, was a previous work they were doing with, I think, 14 genotypes, and they were characterizing different crop varieties. And they saw that when they extracted the parameters of these curves, well, they were not highly related to yield, but they were somehow related to yield enough to make us think that it would be worthwhile to assess to which extent this is helping us to predict yield. Yeah? So this is a a previous study, and then, well, they were extracting basically in a more parametric way to extract the moment in which the maximum slope occurs and other parameters that describe this curve dynamics. Yeah. So now, what are we doing now? It's a project that I think this is the last year of project, so we have actually our last trial is ongoing right now. So we chose different locations that they represent the degree of drought intensity that is relevant for wheat Chilean environments. So we had in the, oh, I always press the wrong button, sorry. Here we have, so we collaborated with INEA. They have their spring wheat breeding program in this location, and there that is more dry and more hot. And in the south, we get less drought, and therefore we wanted to expose this G/E variation. So we had 192 spring wheat genotypes with 3 locations, 3 years, and 2 replicates. And we had some yield components, which is always very nice data, but a lot of work. And some phenotyping, so we were flying with the RGB, thermal, and multispectral cameras. So first, what happens? So when we go back to our schedule, we think, well, the first thing you need to do is to separate this genetic signal from noise. And actually, you see NDVI, for example, that is an index that describes how green the canopy is. You always see, as expected, it goes down because everyone senesces. But also, you have these spatial trends that are time-dependent. So if you measure day 1 and then you measure day 5, obviously, the patterns that you see in the field are correlated to each other. So I think, well, the work by Fred and Diana, for example, his PhD students were also trying to expand on this, on how you can model space and time simultaneously. So in this way, in this work, we were just having like slice per slice in time point, but nevertheless, if you compare this surface, you will see that this part of the field, for example, is a lot more with lower values of NDVI than the rest. And so there is a time dependency in the spatial effects. So then, okay, we get our spatially corrected blues. And then, well, you need to see how heritability changes over time because, well, not only because measurement error changes over time, but also genetic variance changes over time. And obviously, when you are dealing with a trait like stay green, you see everyone at the end gets yellow, and therefore the genetic variance for that trait is decreased. So therefore, the precision you get for the blues is also changing. So it's one of the maybe obvious things to do, but always important to assess when you're dealing with time-dependent data. Yeah? And later, like I said, we can go from this spatially corrected means to something that is more dynamical modeling. And then I will illustrate one of the strategies that can be used, that is splines, not in a parametric way like they did before, but there I wanted to apply this spline method because many times the curves are not so well behaved, so we wanted a more flexible approach to incorporate this time dimension. So for those of you that are new to the world of splines, well, they were, I think this paper says 20 years, but it's actually more years now. But the basic idea is that you have some polynomials that can be linear, quadratic, or cubic, and they are defined in a certain range. So if you multiply those polynomials by a certain coefficient, you can explain your data. So for example, if you have this, you want to explain this data point there, actually that can be predicted from the sum of this spline, this is plus this, and then you add up to that. But if we allow these coefficients to be very wild, they can take any value, and then we perfectly explain our data, and we don't want that, obviously, because we want to be general enough, so we want some smoothing on it. And that's why, well, Paul Eilers and Marx, they embedded this in a linear mixed model for which there is a penalty, so we assume a certain distribution for these coefficients so that we can have some more generality and avoid overfitting. So then I was very lucky because actually Martin Burr, who is Martin? Somewhere, Martin. Martin has just released his nice package, LMM Solver, which I was very happy to see because it made our life a lot easier by fitting these splines. So basically, we took these time points, and here I'm only, so we had the 196 genotype. Here I'm only showing 4 that are contrasting for the dynamics. For example, here we see that in Cauquenes, that is in the central region, we have, for example, this Lasana inea that is higher yielding or higher NDVI, but you have a rank change. So if you see this curve, you see immediately G by E over time, because the position of the curves depends on the location as well. And when we sum, so we said, well, how to summarize, because we don't have clear parameters, like in a parametric model, you can extract the asymptote and other parameters. Here we said, well, probably the area under the curve, so how long are they able to stay green, keeps a kind of summary. So you see there is also, this is a GxE biplot. We see that, for example, in Santa Rosa, that is more further north, this Lasana inea is usually at the top. And in Valdivia, this Genesis is at the top, so it's higher yielding. So there was some considerable G by E for this area under the curve. But well, this is a lot of work, so we want to know if this is really somehow related to yield. So one of the things we did is to look at the same thing, but for yield. And well, one of the things we were happy about is actually the G by E responses somehow coincided, not 100%, but somehow coincided that at least for the environments, the correlations were consistent for both traits. Both traits had a lot of considerable G by E. The G by E was about the same or even a bit bigger than the G by the genotypic main effect. So, well, it seems that there is at least something further to explore with this trait relationship. So later we said, well, actually we will incorporate this on a multi-trait model. We have GBLAP and we want to extend it, incorporating this area under the curve. And there the difficult part comes, or because, well, I was running this multi-trait model and, well, I would have liked this to look a little bit different, but the message or the point I want to make here is that this benefit of including additional phenotypic information is really environment dependent, because here this is a, we see that when we compare the multi-trait model compared to the single trait model, for example, in Cauquenes it works, here is Santa Rosa works a little bit, and the others, yeah, very limited. So, well, this is not what we wanted to see, but, well, that's life. But I said, well, is this really so strange? And I went back to my PhD days. And actually there we had done similar work, but with simulations, and we were comparing simulated data, adding different types of errors and measurement schemes and all sorts of things. And actually there, even though we know the truth, because we knew the crop growth model that was below the data generating process, immediately there we also saw that for some drought patterns, you have, so here the horizontal line means predicting yield from yield, and the points means predicting yield from yield plus something else. And indeed, if you have very few time points, like as represented with the blue or the yellow, sometimes, well, this is the interval between days. So a big interval means few measurements. If you have few measurements like the red ones, it means sometimes, well, you are introducing noise because these curves are not representing well the overall process that is relevant for drought adaptation. So, well, life is always a little difficult, and here the invitation is, I think, well, we are doing a lot of phenotyping in the world. I think this is an area that is expanding enormously, but still I haven't found anyone that has the full recipe to solve it. So I think it's, until now, it's really case dependent. And I think the open question to this team is how we can better understand when it works or not, because explaining it a posteriori is nice, but then we already spent the whole money all the whole time. So it would be nice to find a way to really predict when we should invest in additional phenotyping and how to use it. So I don't have the solution, but I hope we can together move forward. So So to wrap up, incorporating dynamic traits in prediction models can help increasing accuracy and contribute to understanding the adaptation mechanisms that explain GxE. And extracting these summaries are a strategy to incorporate dynamic traits in prediction models. But here I only showed one. There are many others. For example, these multi-trait models with individual time points instead of the summary. We could summarize time with PCA or other indices or parametric models like they did before. For example, maybe we, after all, go back to parametric models. We need to check, although their convergence was not so nice. And obviously incorporating the multi-trait and multi-environment models, I think all the kernel methods that Fred was presenting this morning, for example, could be a nice way forward. So to finalize, I would like to thank the funding agencies and the whole team that helped collecting and analyzing this data. And last invitation, so next year in November, we are hosting, with Charlie Messina, we are chairing the International Conference. This will be the 8th edition in La Serena. It's inter-drought because it's an interdisciplinary approach to drought adaptation. So there are sessions about GxE, modeling, genetics, phenotyping. So I hope it would be nice to see you there in La Serena. The last day there will be a trip to Elqui Valley that is famous for pisco. There are many grapes and irrigation systems, so there is a lot of nice things to do and see. Thank you.
That was fantastic, and I hope we will see a lot of people that are into drought. We've got 1.5 minutes for questions. If there's anyone for Danielle. Somewhere over there? Over there.
Yes, it's a bit intimidating.
A short answer.
So looking at your dynamic curves, your phenotyping course, if you're flying every day, it's quite intense and it's a lot of work.
It was not every day.
I know.
But,
but like what I'm saying is, did you do any kind of optimization of okay, in order to get those curves, we need to fly at least 4 times, and we need to fly every this amount of days in order to optimize your workload.
Yeah, that was a bit what we wanted to do with when we did the simulations. So here. So there, we had the truth, that is the outcome of AppSim, and we added some error, could be big error, small error, different heritabilities. And then we wanted to check a little bit actually how heritability changes when you modify your measurement frequency. So what we learned there is actually if you can improve your curve estimation in 2 ways, either having perfect measurements, but that's never true. You can also add more measurements. So there is a sort of trade-off between the heritability at single time points, so the precision you get for each time point, and how often you measure it. So if you have a small heritability, then you definitely need to measure more often. But that's really, trait to trait dependent. So I cannot give a general rule. But there are some colleagues from Universidad Talca, they wanted to measure every hour to make an hourly thing, but that was more to characterize canopy temperature and stress responses in a single day. But it's work that is coming.
Okay, thank you. Thank you, Daniela. It's fantastic.